everyone. Welcome back to BEaTS Research Radio at the University of Ottawa Heart Institute. And for the time being, we're going to be streaming from the comfort of my home office here in Ottawa. And today we have an interviewer, is Dr. Manisha Kulkarni from the University of Ottawa, from the School of Epidemiology and Public Health. Dr. Kulkarni research is quite interesting, actually unique in the sense of she's one of those like myself, a interdisciplinary scientist that he was training in one field, but she's now working really actively in a completely different field that we're going to learn more about this. So Dr. Kulkarni is, she has received many awards, including the Early Research Award from the Ontario Administrative and Innovation, and also she's funded by the CHR, NSEP, PHC, and IDPRC. Her research is around understanding public health and epidemiology and how that is affecting and actually influencing our health and also pandemics as the one we're living today. So thank you so much, Manisha, for being here today with us and for taking the time actually for doing this with the BEaTS Research Radio.
Yeah, my pleasure.
So I want to actually start by if you could actually tell us a little bit of how actually this happened, because you're you're an entomologist and now you're working a little bit in the epidemiology side. How this happened to you? How actually you felt that it was the right move or not?
Mm-hmm. So actually, the two fields aren't totally distinct. So my training is in medical entomology, so studying insects as vectors of disease. I originally studied environmental biology and specialized in parasitology. And many of the parasitic diseases are actually transmitted by insects and other arthropods. So in my graduate studies and my PhD, I ended up specializing in malaria and working in East Africa and Tanzania on malaria ecology and epidemiology and control. So my studies spanned biology and ecology, but also epidemiology and parasitology. So right from the beginning, I was multidisciplinary, and I've managed to continue this into my work to date, expanding from malaria research to other vector-borne diseases and other emerging diseases as well.
So when we speak about vectors, so what is a vector itself, like in the definition, actually, for understanding better?
So when we talk about a vector, we talk about something that can transmit a pathogen from one organism to another. In the sense of vector-borne diseases, these are insects or arthropods that propagate disease. So they're able to pick up a pathogen from one host and transmit it to another host. But one of the important features of a vector is that there's actually development of the pathogen within the vector organism itself. So it's not just a mechanical carrying of pathogen, it's actually involved in the developmental life cycle.
So in that sense, we have the impression because of the weather we have in Canada, right? Because we have long winters and in most of Canada. We have known that the sort of vector disease, but I'm quite familiar with Lyme disease. Mm-hmm. From reading your website last night, I actually was quite intrigued about the research you're doing with Lyme disease and how that is also extrapolated to different regions in the planet, like South America, Africa. Can you tell us a little bit about that?
Absolutely. So one of the focuses of my research here locally is tick-borne disease, and Lyme disease being one of the main ones. And we've seen really an expansion of Lyme disease and northward range expansion over the last decade because of an expansion of the tick populations due to things like climate change and warming temperatures that are allowing a longer growing season. So the ticks are able to survive and reproduce at higher latitudes. But we're also seeing that there's much more human contact with ticks due to urbanization, landscape change, and things like that. So it's quite complex in terms of the different environmental changes that can affect vector-borne diseases. And of course, insects and arthropods are very sensitive to environmental conditions. So any change we have in the environment can impact disease epidemiology.
So this comes along that everything is connected at the end of the day, and I think that this is no exception, clearly, right?
Absolutely. And when we're talking about global emergence of disease, we have to remember that the world is so connected these days. So something that emerges in one part of the world can quite easily cross borders and affect populations in another area. So one of the areas of my research in mosquito-borne diseases, for example, we're studying Zika virus. But it's not just Zika virus. The same Aedes aegypti mosquitoes can transmit dengue and chikungunya. So in Latin America and areas of South America where I'm working with partners, we're looking at the co-circulation of all of these different arboviruses to see what are the impacts and how can we really implement effective control strategies that would tackle a range of different diseases, not just one single disease.
So in that sense, the mobility of the vectors of these ones are of the entities that are transferring or moving the disease around. In this particular case, we have been actually calling them that insects of arthropods, as you say. But is that something that we can extrapolate to the human behavior when we are actually, like being vectors of carrying a disease, and we can transfer that? Is that something that it could be extrapolable to the human disease when the human is the one transferring the disease?
Absolutely. I mean, when we have diseases where the human is the reservoir, as we do with some arboviral diseases, malaria, for example, or respiratory pathogens, as we're seeing now with COVID, we need to be mindful of how populations are moving and mixing, potentially introducing new pathogens to immunologically naive populations, which can lead to epidemic situations. A lot of the dengue situations that we're seeing in Latin America are the reintroduction of new strains to which the population has no immunity. So if we're able to better understand how populations are moving, both internationally and locally within a given area, there's seasonal migration of people, for example, we can better predict when epidemics are going to happen. But we also have to be looking at the whole environment. So looking at the human population, the vector, the environment, and in some cases, the wildlife population, which could serve as a reservoir for some pathogens.
So when we say that, what I'm trying actually to better understanding here is that, we have been seeing a lot of these models, right? Epidemiology models to try to understand the progression of COVID, particularly in the setting we have now.
Mm-hmm.
But all this is built on the basis of probably having different species as models of this vector transmission. Is that correct?
Yeah, so one of the aspects of my work is to develop spatial models. So we're able to look at the factors, environmental factors, sociodemographic factors that are associated with the presence or occurrence of vectors in the environment and the propagation of pathogens. And we can then effectively map those and see where are the areas that are at highest risk for disease emergence, or where are we likely to find the populations that are most at risk that are living in areas with vector populations in conditions where we have good environmental conditions for pathogen transmission, for example. So our models aren't necessarily the dynamic mathematical models that you're seeing with the epidemic propagation of COVID. Some groups work on that, but my group, we work on spatial distribution models. So really, what are those factors in the environment that are able, that allow us to predict where populations might be most at risk?
This is fantastic, actually. What we're going to do, we're going to take a brief break, and we'll be back in 2 minutes to learn more about, your interesting research. So stay tuned. We'll be back in 2 minutes.
what are the non-modifiable risk factors for cardiovascular disease? Cardiovascular disease can be caused by a variety of risk factors we cannot control. The older you are, the more prone you are to chronic conditions like heart disease or stroke. However, men have a higher risk of cardiovascular disease earlier in life, while women are more at risk after menopause. Women may also experience different symptoms of heart attack that may go unrecognized. You may be more at risk depending on your ethnicity. People of Asian, African, or First Nation descent have higher rates of cardiovascular disease. Your ethnicity may also influence your diet, which can put you at an increased risk for cardiovascular disease. If you have a first-degree relative who has had cardiovascular disease or a stroke before the age of 55 for men and 65 for women, your risk of heart disease doubles, and regular screenings with your doctor are recommended. But there is good news. Up to 80% of premature cardiovascular disease can be avoided and more effectively managed by taking these proactive steps. Quit smoking, be physically active, eat well, achieve a healthy weight, manage your stress and any anxiety or depression, and see your doctor about managing your cholesterol, blood pressure, and blood sugar levels. For more information on cardiovascular disease, please visit our website.
Welcome back, and we are having today in today's episode of BEaTS Research Radio, Dr. Manisha Kulkarni from the University of Ottawa School of Epidemiology, and she's telling us about her exciting research in vector-transmitted diseases. And I have a question follow up with what we were briefly discussing in the first section, and it's about if the environmental impact of disease transmission, and if that really has an impact when we're thinking about what is going on today with COVID, and if environmental conditions like pollution, temperature, it could have an impact on how the disease is transmitted, and if that is something that you have explored, not probably in COVID, but with other diseases.
Mm-hmm. That's a great question. Environmental conditions can have an impact on all components of what we call the epidemiological triad. So we have host, pathogen, environment. And environmental conditions like air pollution, for example, could create susceptibilities in the human host population that would make them more vulnerable to infection, for example, with underlying conditions. There's a lot of research going on right now with COVID in terms of the impact of environmental conditions. We don't know whether there's going to be seasonal variation due to temperature and humidity, for example, that can affect some viruses. It's not clear whether this is going to be the case, and we're going to see a seasonal attenuation with COVID transmission. In terms of the pathogens that I'm focusing on, environment has a major role to play because vectors themselves are cold-blooded, right? So they're impacted by temperature changes. So anywhere that there's a warmer temperature, they're more able to survive. They can develop faster into adults. They can actually digest a blood meal faster, so their biting frequency increases. But also the development of the pathogen, whether it's a virus or a bacteria or a parasite, is actually sped up in warmer temperatures. So you have all of these things that are contributing to almost an exponential increase in transmission potential at higher temperatures, which is why we see a lot of vector-borne diseases in the tropical areas of the world. So if we have global warming, temperature change, we are seeing this kind of range expansion of vector populations, and putting populations that are susceptible, have never been exposed to disease, and don't have immunity at higher risk. So in East Africa, for example, we're seeing that malaria is moving up in elevation. And this is where we have really densely populated areas in the highlands. Many populations have not been exposed to malaria. So there's potential for disease outbreaks in these populations. We're seeing it in Latin America as well, with the Andes, malaria moving up to higher elevations, but also other environmental conditions like with urbanization, we have a lot of poor sanitation, crowded populations living in poor conditions, and these are great breeding areas for Aedes mosquitoes that breed in discarded containers, for example. So the environment can have so many different impacts on propagation of disease, whether it impacts the host the human host, the vector, or the conditions for which disease can be propagated.
So, in the sense that when we're trying to actually to develop these models, you know, like the epidemiologists and the mathematicians and all the experts in the field, they're coming up with these models of, you know, of prediction models, right? So I'm wondering if there is an effect of, you know, the truly ethnicity distribution of the population, in the sense, for example, we are comparing, you know, the population we have in Toronto, which is I think 75% of the inhabitants in Toronto, they weren't born in Canada. When you're comparing to a more, you know, narrow distribution like in the West Coast, and where, you know, mostly, you know, Canadian born, second generation, third generation. So what do you think of that? It could actually have an impact, because if that is the case, when you're trying to, you know, use the model we're using in North America, try to extrapolate to South America, where the mobility within the countries is much more limited, and in that particular case, we are not accounting for an important factor in that.
Absolutely. I think all of those factors are important to consider in terms of the differences in the host population. You know, there's differences in age distribution in populations. You know, we can't really compare the situation in Italy to the situation in China because of the different age distribution of the population. What I'm really concerned about is what's going to be happening in Africa when COVID is really starting to take root. There we have a much younger population in general, but the health system is very poor. So it's really hard to extrapolate from one population to another. And I think that all of these issues really need to be having more research. We need to be able to have better parameters for these models so that we can really more accurately predict what's going to happen. But that takes a lot of time. It's amazing how quickly the research has evolved on COVID. We saw the same thing when Zika came out. Fortunately, that, you know, didn't last as long and didn't have as huge a global impact. But it's an area that we need a lot of research, and I know a lot of people are working very hard to get those answers.
So in that sense, I believe that this is a good opportunity for us to learn that, you know, good research takes time, and we cannot rush into this. We cannot rush into conclusions. We cannot rush into, you know, like saying this is the way it goes, because research is fluid. It's something that evolves with time. The conclusion of the results with the research we're doing today, probably tomorrow they're going to be different, but the data is always the data. The data is the only thing valid we have there. And you know, what lessons do you think we should be able to learn and you know, from this pandemic? And also, I would like to see if you can share more about your exciting research, because, you know, like, you know, COVID is is the pandemic we're having now, but, you know, you never know what's going to happen in the future. And I think we have to use this opportunity to learn new lessons and to really put more attention on predictive models, on emerging diseases and vector-related diseases as well, right?
Absolutely. I mean, I think, you know, this pandemic, it really drives home the point that the way that humans are interacting with the environment, with wildlife populations, is causing diseases to emerge. People have been looking at coronaviruses and emergence of these diseases, you know, since we had SARS. And we know that there's spillover of zoonotic pathogens, that more than 60% of emerging infectious diseases are zoonotic. They come from wildlife. Yet people aren't changing the practices that we have in terms of interacting with wildlife. There's zoonotic spillover of transmission with West Nile virus and with Lyme disease locally. With urbanization, we're putting people into contact with enzootic transmission cycles. The opportunities for people to become infected with new pathogens are just increasing with increasing population density in areas where we have high biodiversity. And it really draws attention to people's ability to really predict where diseases are going to emerge. We may not know exactly what the pathogen is going to be, but we do know that if we keep continuing to interact with the environment and with wildlife in this way, we're going to have more and more disease emergence events. So, you know, my work really tries to highlight some of these upstream drivers of disease emergence, because we can predict where those are going to happen, and then we can focus our surveillance efforts there. We need to be able to strengthen health systems and surveillance systems in areas where we're going to likely have disease emergence, so that we can detect it early and not have global spread so quickly as we've seen in this latest event. But really, vector-borne diseases are one of the main concerns for for new emerging diseases. On the one hand, we have disease emergence and new pathogens, but on the other hand, we still are tackling a lot of these endemic diseases like malaria and dengue and other neglected tropical diseases. So we can't take our eye off the ball there. We need to be working on both fronts to tackle the endemic diseases, but also keep an eye out for the new emerging ones.
And I think that with all this of the pandemic and the situation we're living these days, I feel that many students, young, the next generation, the youngs and the youth are maybe thinking about pursuing a career in science, because science is extremely important. We do need more scientists and really passionate scientists. What will be from your a message to them if they want to actually get into the epidemiology or your particularly your research? What will be the one tip you will actually give them?
Yeah, absolutely. I think pick a field and pursue it. And there's always opportunities to branch out into other fields. I think it doesn't matter if you start in epidemiology and then learn more about entomology, or if you want to start in entomology and then learn about clinical science. Pick a starting point and go from there. There's so much goodwill amongst scientists to work together in multidisciplinary teams. I think it's the way of the future. And I think the future is really, really bright for science and for doing epidemiological health-related research. So absolutely, I'd encourage people just to pick a starting point and go from there.
And within Canada, so like, what is the project you have in mind for when things actually are back to normal? Because you're working in the field, right? So you're working. I saw some wonderful pictures in your website. So what is the next project you're working on?
So we have a variety of projects here in Canada. We're working on tick-borne diseases, doing surveillance to see where the risk is highest in populations. We're working with different neighborhoods in Ottawa, for example, to look at what are the impacts of different urban planning practices and urban development on Lyme disease risk. How can we better protect people that are living in these areas that are bordering natural green space where the risk is highest? So we have some intervention projects working with the National Capital Commission within Ottawa. We're collaborating closely with Ottawa Public Health on these projects to really have locally adapted intervention strategies, potentially that we could implement for populations at risk. Abroad, I work in Tanzania and East Africa. We have a bednet trial looking at novel formulations of next generation bednets to combat insecticide-resistant malaria vectors. We're still seeing a resurgence of malaria in a lot of areas, and we need new tools to combat that disease. And then in Latin America, I'm working with partners in Ecuador and Colombia and Argentina to identify entry points for controlling Aedes mosquito vectors that transmit Zika, dengue, and chikungunya. So a lot of field work is going on. It's been on pause now, hoping to get back to it soon as much as we can. But there's just so much going on right now. It's interesting times, and I think we're all just having to adapt the best we can.
Oh yeah, absolutely. And I think that, as you say, it is something that resonated on me, is that when we collaborate, we're stronger. And I think we're learning. This is a time for scientists and for the community to learn from each other, to learn that it's a good thing not to know something, because it is an opportunity for us to learn and to actually become better and collaborating. This is what we live in today. And, most of the diseases you were actually discussing with us, the solution is not going to come from one scientist, but from a team. And I think most likely the solution will come from the next generation of scientists. And I hope that, the youngs and watching this, they will get that message that we need them. We need the actually passion and to inspire others and the next generation that is coming. So Dr. Manisha Kulkarni, thank you so much for joining us and taking the time of being here with us. It was truly a privilege. I'm really excited actually to learn more about your research. And we're going to have your website in here. It's going to be showing up in the screen. And I invite everyone actually to learn more about your exciting research, and congratulations. It's really actually inspiring to see a truly interdisciplinary scientist, someone who was trained in a different field doing this amazing work that is really resonating within Canada and around the world, because working with developing countries is the most Canadian thing we can do. And thank you so much for your time. And we look forward to learn more about your research in the future.
My pleasure. Thanks, Emilio.
Thank you.
Bye.
And everyone, thank you so much for being here today. And it was a pleasure. And thanks again to University of Ottawa Heart Institute, the Biochemistry, Microbiology, and Immunology Department, eLife, and the Canadian Biomaterials Society, Ottawa Student Chapter. And see you guys next time. And stay tuned because we'll have more exciting episodes to come. Have a good day. Bye.
BEaTS Radio, scientists interviewing scientists. Canada is a worldwide leader in science and innovation. However, our communities need to be more connected to current scientific research in order to drive the future of scientific innovation. So what steps can we take to more actively engage and connect our community with scientists and vice versa? Innovation and discovery are at the core of the current efforts of our government, but science sometimes gets slightly complicated to understand. At BEaTS Radio, we will tackle this by having young scientists and early career researchers interviewing scientists and innovators. By dissecting science in simple terms, this will allow our community to better understand the importance of science for our country.